Hello there. I'm Patrick Mills. I'm obsessed with amino acids. I've been studying them for 20 years and I'm here, and I'm a research scientist. I've been doing this for some time, of course. I'm here to tell you how we've cracked the amino acid code. Okay? So, it's been an obsession. Okay? There's me, there's my lab. I'm actually from Chicago, and that's my lab in Chicago. And we love to talk to anglers, so feel free to check out our website and feel free to also email us. Now, let's talk about amino acids. What are we going to talk about? Well, we'll keep it simple to start. We're going to talk about what exactly an amino acid is. And once you've figured that out, we can move on to how fish detect amino acids, which to anglers is a very, very, very important question. Answering that is kind of a crucial thing. Okay? Once we've figured out how fish detect amino acids, we can then go on, which is the most important part of the talk. How can anglers then go and use amino acids to catch more fish? Now, amino acids are fish feeding stimulants. They'll make fish eat food. And that's the product we have developed. Okay, we'll talk more about that later. And there'll be a little bit of time at the end for any questions you guys have. All right. If anything really jumps out at you, feel free to shout something out. I'm happy to answer. Okay. So first up, what's an amino acid? Well, I'm going to take you back to, hope you don't have a flashback, take you back to biology class a little bit. Okay. It turns out that proteins, our muscles essentially, are made up of building blocks we call amino acids. And there are 20 amino acids in nature. And they all string together like Legos to make larger structures we call proteins. All right? So think of aminos like Legos. They stick together to make proteins. Okay. That's a little bit busy, but it's important. Okay? Turns out there are 20 natural amino acids in nature, 20 building blocks for proteins, and they come in 4 basic types. And they have names, which we don't really need to get into, but there are 4 families of amino acids. That's important for reception, which we'll talk about in a moment. So the take-home message is, proteins are made from amino acids, and there are 4 simple types. Okay. My hero. I was obsessed with amino acids because of this book. Okay, if you are as old as me and you read Carp Fever back in 1984, it changed my life, and I've been obsessed with amino acids literally ever since. And Kevin's book, Kevin Maddox, he listed the amino acids in order of potency. So he did some wonderful, really good testing in his house back in the 1980s. And he found out that valine and lysine were the most potent amino acids. They gave a very good feeding response, stimulated fish to feed. And then he ranked everything else, kind of like a league table. So if you like, might be controversial, lysine is Liverpool, valine is Manchester City. We go all the way to the end there. Cysteine may be Yeovil Town. Any Yeovil Town fans? Never there. Okay. Now, that was brilliant work done back in the 1980s. Bang up to date, we revisited Kevin's work, and that's what we found. Remember those 4 families? Okay. Turns out that lysine and valine are members of the basic and neutral families, and they are the most potent fish attractors. If you look at the color coding of the chart, it's Kevin's chart repeated, you can see that the most potent amino acids, in other words, the best fish attractants, are either belong to the blue basic group, lysine is the king, or the purple neutral group, valine is the king. So you can use literally any blue one or purple one to make a good fish feeding attractant. Okay. Now, one or the other, or not both. Okay? Kevin found out something very interesting. If you read Carp Fever, maybe we'll touch on it later, but they work individually but not together, which is interesting. And that's a simple fact we've been working on for 20 years, which has allowed for the development of what we call the BVRM, the bivalent receptor model, meaning 2 amino acids are involved. That's a bit more detail I don't want to go into, but it's out there. Okay. Now, there's a weird picture, right? So all living organisms, including these living organisms, break down amino acids and then rebuild their muscles with new amino acids from the food you eat. Okay? And these guys have made a business of this, of course. If you're a professional bodybuilder, you have protein amino acid supplements which rebuild your muscles after working out. Okay? So muscles are always constantly regenerating. They're always being broken down through work and exercise and then regenerated through growth. And this is a continual process. Now, if you're as old as me, you'll remember this. Only fools and horses. Classic episode, if you remember this. Trigger, who of course works for the council, he sweeps the streets. And he has a broom, and he's had it for 20 years. And he says, classically, I've maintained this broom for 20 years. This old broom has had 17 heads and 14 handles. So obviously it's not the same broom. And that's kind of how regeneration works in animals, right? So brooms regenerate, well, the heads every 0.7 years if you do the maths, and the handles every 0.8. Turns out humans, all our cells, turn over every 7 years. So you're a different person now than you were 7 years ago, completely different person, because all your cells have regenerated. Kind of interesting, right? Now, why is that important? Because all animals are breaking down and building up. And the waste products, those building blocks, amino acids, are being excreted all the time. Okay? Humans are excreting amino acids all the time in the form of fingerprints. Everywhere we go, we leave fingerprints, which have our amino acid profile inside our sweat. And you can actually develop these amino acid fingerprints with something called an anhydrin, which is what the FBI does, all right, for fingerprints. So fish are just the same. Fish are excreting broken down proteins, amino acids, all the time, which is really a, hey, I'm here signal. And that's what ancient fish did to find each other. They used amino acid trails, which you can't hide, okay? You can't hide because you're always excreting aminos. Now, this is where it gets a bit weird. Now, these are just pictures from the internet. I haven't done anything bad to any fish, right? So no fish were harmed in this study, okay? But we've got to know how fish detect these amino acids which are in pond water. So think about it. Creatures, food, worms, mussels, whatever it is, they're excreting these amino acids. These amino acids are then smelled by the fish and they can hone in on them, okay? So the question really is now moving to part 2. How do they do that? How do they detect amino acids, i.e. food signals? Well, you can see there the nose or the nares of the fish. It's a two-way system. Doesn't really work with a laser, but the big hole is where the water goes in, and then there's a baffle or a kind of a block, and it comes out the back. So it goes in, circulates, and comes out. This is the gory slide. This is not me, this is internet. Right? So if you were able to peel back the layers of skin of the fish, you would find these kind of, I can walk over actually, these tracks here, these tubes, they're called olfactory tracks. So it's a tube which the pond water goes down, and it gets to these big old bulbs down here, which are called olfactory bulbs, these kind of bigger bulbs here. Okay, and that's where olfactory reception happens. Okay, so olfactory reception, smelling. Okay, if you notice, the olfactory bulbs are actually almost the same size as the organ behind them, which is the fish's brain. Okay, so the fish's nose receptors are almost the same size as its brain, and that's true for virtually every fish. So they're so reliant on this technology, so to speak. Now here's where it gets interesting. This is an electron microscope picture of what's inside those olfactory bulbs. Okay, so it's a bit like a pomegranate. There's these little seeds inside, which are called olfactory rosettes, and there they are. Okay, black and white, of course, because it's an electron microscope picture. And as you can see, it's kind of a spherical object, and on the inside it looks like kind of ripples. Those are ripples of skin. If you think about your gut and how it absorbs food, the surface area of your gut lining is wavy, like the tines of a comb, to increase surface area. And that's exactly what's going on down there. And those little folds are called the olfactory epithelium, which literally means olfactory nose, epithelium, skin. So it's a skin surface where the receptors are placed. Okay? If we zoom in one more time, very interesting picture. So we're sitting on one of those skin surfaces, And we see these anemone type structures. So if you see like an anemone in a rock pool, it's kind of like that, right? So the anemone is a nerve cell, okay? So the center root there is the nerve cell, and those fingers that come out are called cilia, and that's where the receptors are. So we've kind of zoomed down all the way to where the reception happens. So the reception happens on those fingers, all right? That's quite a lot of information, so we took you back to biology there, but hopefully not too bad. There's a zoom in. So there's the starfish or anemone cilia, okay? And now we're at the micron scale, a millionth of a meter in size. So we can't zoom in anymore with a microscope, so it's kind of an image kind of drawn from now on. And I've taken the trunk of the cilia and I've stylized it. The black hexagons, and this is where the action happens, the black hexagons are amino acid receptors, the spots where the amino acids from the pond water, which is out where we are, come down and sit on those cilia, on those black hexagons, and cause a signal. Okay? And they're in clusters. So that's stylized, but the receptors are in clusters, and binding to those receptors causes a food here signal. And that's how fish detect amino acids. So we've kind of gone through and answered our second question, but not completely. Now this is where the good stuff starts. So thank you for being patient and, you know, working through the essential physiology, we call it. Okay. Now it turns out that the amount of amino acid detected is either not enough, so think of Goldilocks and the Three Bears, too cold, just right, perfect, or too hot. Optimal stimulation comes when you have a just right coverage. So let's look at what that looks like. Bit of an animation here. So we're zooming in on the cilia. There's the receptors. That's a too cold coverage. If you see there, we're less than 50%. So 2 out of 9 spots covered. That's a too low signal. That won't catch you a fish. It's a too cold signal. So if you've got less than 50% of your receptors occupied, too cold. If you got that concept, you're good. What do you want? You want just right. A just right coverage is between 50 and 85% coverage. And this causes optimal stimulation. This is what catches you fish. But how do you achieve that? Well, we'll talk about that in a moment in more detail. But basically, that's what we call the therapeutic range. So if you know anything about like drugs and pharmacology and things like that, it turns out that drugs are designed to operate between 50 and 85%. That's how they work. And it seems to work the same for amino acids as well. Okay. Too much. This is very, very easy to do. It's very, very easy to add too much amino acid to the bait. If you cover every single spot, overstimulation, that's essentially what we call an overdose. So that's a technical term, it sounds bad, but just think of like 10 cups of coffee, you don't feel so good, you just want to lie down, right? So that's not optimal for fish if you have an overdose situation. So the take-home message is, if we're using single amino acids like Kevin Maddox did back in the 80s, between 50 and 85% of the sites covered is what you want. Okay. Now here's the big problem. So if you think about it, how do we fish? So we may perhaps dip a bait in some neat amino acid powder or something like that. And then you cast it in. And what happens, you start from zero and then the amino acid starts to dissolve and you get a ramping. So the amino acid concentration goes up and the number of sites starts to fill proportionally, okay? So, just read it there, as the AA concentration increases, so does the number of occupied sites and so does the response. So the fish get increasingly more turned on, the more amino acids there. Just to remind ourselves, that's the too cold, so that's when it just starts to dissolve. We quickly, and we'll talk about this more in a second, we quickly go through the just right, which is what we want, and then unfortunately we usually end up at too hot or too much. Okay, now this is what's been the bane of amino acids since the 1980s. So anglers have noticed that when they use amino acids with their baits, sometimes they catch, sometimes they don't. Amino acids have been extremely inconsistent. Why is that? Because either they're too cold, or too hot. The optimal stimulation range is extremely narrow, okay? And I put some hard numbers there from our research, right? So you're looking at 1.2 milligrams per minute of amino acid, which in terms of a real kind of understandable number is about 0.1 of a grain. So imagine 0.1 of a grain of salt per minute dissolving into the water. That's how low you have to be. A little bit further either way, you're too hot or too cold. So it's way down there in concentration, super low, and it's very narrow. So you can see how hard that is to establish, right? So you can hit it occasionally, almost by luck, and that's when you catch a bunch of fish. But typically it's too hard to achieve consistently, and that's always been the problem. And that problem has taken 20 years to solve, and that's why I'm here today. Okay, so is it mission impossible? How can we get that perfect 50 to 85% consistently? If we can do that, we are in business, okay? Newsflash, this is why I'm here today. We've managed to solve this problem. It's 20 years in the making, but here we are, okay? So Impulse, that's the product we've developed. We actually lock in the optimal coverage. So what we've got there, we have a mixture of white spheres, which are amino acids, if you remember from a previous slide, and now new things are the green crosses, okay? The green crosses are proprietary blocking molecules. What's a blocker? A blocker is just a molecule that gets in the way. So it occupies the site, but doesn't stimulate it. It's like putting a piece of chewing gum in a lock, or a car lock or something. You can't open the lock because there's a piece of gum there. And if we use a 50-50 mixture of blocker, the crosses, and amino acid, the circles, look at the coverage. It's exactly what you want. So here's the key finding we found, right? It's not about concentration anymore. That narrow range is no more a problem, because we've locked in the coverage through a ratio. If you understand that ratio issue there, how we've locked in 50%, say, by mixing a 50-50 mixture of blocker and amino, we're good, okay? And that's the background of why Impulse now, our commercial product, is consistently stimulating fish over a very broad range of concentrations, okay? Now, the fun stuff. So that was your kind of science homework completed. So thanks for being patient there. We are now going to look at some kind of stuff you can kind of use. Now, I, of course, work in a lab. I work in a small college outside of Chicago where I'm a chemistry professor. I have a research team of students. We have 3 research tanks up there on the right, and I'll talk more about how they're set up in a second. But the fun stuff, bottom left here, we actually see fish being exposed to an Impulse solution, okay? And this is now a consistent result. We see this every time we use our stuff. So that's what we call involuntary feeding, okay? We have amino acids coming out of the kind of the small object on the right there. You can just see some, we've dyed them black just because you can see them. We don't dye them every time, but when we want to see them, we dye them black. And it happens with or without dye, so it's not the dye. And as you can see, they're digging around. And that's what you want. You don't want them going crazy. You want them being kind of actively searching. And that's the ideal feeding response. Okay. It's very similar, if you read Carp Fever back in the 80s, to Kevin Maddox's original experiment. Kevin Maddox simply fed in a solution of amino acids through a pipe into some gravel and then observed the fish. And that's essentially what we do. We're just a bit more sophisticated. And Kevin's experiment was genius because when you put the amino acids into the gravel, you're starting at 0 and then slowly increasing the concentration. So you do go through the 3 phases, too cold, just right, too hot. And then if you saw the carp respond quickly, that was the most potent. So that was valine and lysine. Okay, and then he ranked them in order, which was fantastic. Okay. Our experiment, a little bit more sophisticated. We are using a doser, which is essentially, you can see it on the right there, a kid's sippy cup with a diffuser, which is an air stone from an aquarium. Cover that with gravel, and then the pipe runs out to a reservoir which we dribble in about 200 milliliters of amino acid solution. So the solution comes in, it diffuses out, and if you notice, it says, Harry Potter fans take note, Godric's Hollow, right? See if you remember that. It's a little indentation. So I've actually got in the tank, I've dug out a little indentation in the gravel there, and if you look, it's doing it right now, the aminos are just dribbling over the edge and collecting in the hollow, right? So you can imagine that happening in the wild, but the doser is actually your bait. Okay, and the fish are coming and digging up the gravel. There's no food signal there, right? There's no flavor, it's just amino acids. The fish are involuntarily stimulated to feed. They're actually mouthing the gravel quite strongly there. Okay. That's my proof. Okay, so I have tank proof. But what happens in the tank is different than what happens in the wild, of course, right? So we've sent it out for testing. It was probably 2013. We sent our Impulse prototype at that time out for testing at Sparshot College. It was tested by 2 students in 2 different projects. First was Tom and then was Ollie, and they tested Impulse. Tom was a match angler style, so it was anglers fishing alternate pegs with and without Impulse dose baits. They got a 2-to-1 catch rate differential as shown by the pie chart. And then Ollie came a year later. He used Impulse dosed boilies, which were very porous because you have to have the stuff come out. We can talk about that more later if we need to. We had Impulse dosed carp baits, let's just say that. And he also got a 2.1, slightly better catch rate, but they were fishing like 1 rod with and 1 rod without. And you know, that's the technique we've tended to go with because carp anglers love to fish 2 rods, of course. And most recently, this year, if you're a big fan of carpology, you may have seen the video. We had Luke and Ian take Impulse out to a runs water, 1 rod with Impulse, 1 rod without, and their catch rate differential was 13-to-1. And you can actually watch that on carpology. If you're more into match fishing, Mark Sawyer, wonderful human being if you've ever met him, awesome guy, right? Mark Sawyer went out, fished more of a match style, so it was method feeders with micros dosed with an Impulse solution, angling times, and Mark calls it like, you know, a Jeopardy test because he just turns up and fishes 1 time and the camera just rolls and people show up and they watch him, so it's completely unedited footage actually in both trials, so you can watch it. And he recorded an 11-to-2 ratio. So obviously much better than before, but you know, we've improved the product. That's, you know, that's a really good result in both cases, but that's what they got. That's what they got when they did their test. And you can look at each of those magazines' YouTube channels for the videos, or actually, and I'm gonna show you my guide here. We've actually written a user guide. There are links to those videos in the user guide, and you know, if you want to come and grab one at the end, feel free, they're free, or you can stop by the booth. We have a booth today and you can grab one there. Okay. So common questions, right? So I'll throw it open to questions and answers in a little bit, but I'm going to go through a few classics, right? So first things first, it's more of a statement. Impulse needs to get to the bottom. Okay, so we have a liquid product which soaks into absorbent baits like pellets, and then they go to the bottom and release. We have a gel which you coat harder baits like boilies or maize, sinks to the bottom, disperses. It's that Godric's Hollow effect, right? Okay. What you don't want to do is create a sloppy spod mix. Okay. You can use your dosed pellets, your dosed maize, whatever, in a spot mix for sure, but the last thing you want is a soupy spot mix because you'll just make a cloud at mid-water. You need it to get to the bottom. Okay, so general spot mix is fine, just not sloppy ones. Okay, PVA bags, by far, in my opinion, are the best application. Okay. Now, as we mentioned, we showed you the pictures earlier. Impulse needs to establish a minimum amount, a minimum concentration, so you get past the too cold region. Okay. That limits you a little bit in terms of boilies. Okay, so oftentimes people ask me, can I put Impulse into my boilie base mix? And that's a yes and no answer. If you're making a standard boilie, which is kind of hard with a firm kind of skin, literally the aminos won't be able to get out quick enough. Okay, so a standard boilie, you can't put it in the base mix, unfortunately, because they're trapped inside. Okay, in that case, you would use the gel and surface coat them. However, if you have a porous boilie, which we tested at Spa-Schult with Ollie, or a soluble boilie, yes, you can incorporate Impulse into your base mix. Next question, you're thinking, well, why is the picture of a fjord there? Well, you've seen these pictures on TV where people have these flavored liquids and they put some boilies, some hook baits in a jar, and they're dosing them with this flavored liquid for months, right? And then they cut them open and the liquid's soaked all the way through the core of the boilie. Looks great, right? And it is good, it's good, right? But unfortunately, that when you put them in the water, if it takes 3 months to soak in, it will take 3 months to leak out, right? And it kind of reminds me of my trip to Norway. If you know anything about fjords, there's always the same question when you're on the fjord tour. How deep's the fjord? And the guide will tell you the fjord is as deep as the mountains are tall. Why am I telling you this? The time it takes to soak your solution in is the time it takes for it to leak out. So standard long soaks don't work. You have to surface coat and have it get out quick, okay? So a couple of quick caveats there. Base mix is okay for a soluble or a porous boilie. Surface coating, not long soaks. But otherwise, you can use it with boilies pretty effectively. Impulse is made of amino acids. Amino acids are digested by microbes, right? So it's a food product. Out of the bottle, it becomes a food, right? In the bottle, it's actually pretty concentrated, so you can't have microbial action in a very concentrated solution. It's a bit like honey in that respect. But once you get it out, if you dilute it especially, you got between 12 and 48 hours to use it. And our kind of like main kind of caution to anglers is use it as quick as you can because it is a perishable food item once it's let loose. Okay. All right. A couple of other quick things. Obviously, this has been developed into a product. It's called Impulse. We have a booth. You've probably seen it already. And we have the liquid, the paste, and the gel. And we have a wonderful website where you can, get this stuff when you're not at the show. All right. I'm a nerd. That's a joke. Not a very good joke. All right. That's me in my lab. Be sure to stop by the booth. Pick up a free guide. I've got a few here. And, product placement. This is kind of what we've got at our booth if you're interested. Okay. Any questions? Don't be shy.
Is that a liquid form of worms have become the new fashion?
Yeah.
Everyone seems to be fishing with worms now. So is that a liquid version of worms?
It's a great question. So worms and all living organisms are excreting these amino acids like we mentioned, and they're excreting a profile of amino acids. So all of them, all 20 amino acids in slightly different amounts. The interesting thing, and we thought Impulse would not work with chopped worm, for example, but it works actually very well because amino acids should not be mixed with Impulse because it causes side effects, it causes cancellation, it's not as effective. However, it turns out that the concentration of aminos in natural foods is actually very low, and we got reports back from an angler who tested Impulse with chopped worm and it worked great. I actually thought it wouldn't work, but it turns out that our concentration is 10 times higher than what's actually in worms. So the answer is you can use it with naturals, particularly chopped worms.
As I say, chopped worms seem to be the new go-to sort of
Yeah.
If there's an alternative, that's
Yeah. If it was me, I would probably soak some breadcrumbs, some micros or something, and put them in with the chopped worm. So it could carry, because remember you've got to get to the bottom, because chopped worm can make a cloud. You don't want a cloud, you want to get to the bottom. So we're good. All right, a few more minutes. Question?
When adding them to like PVA bags, is it a case of like kind of less is more sort of thing?
Fantastic question. So when we worked with Carpology, Luke and Ian, they actually did it perfectly. And if you look at the user guide, on the back, it's kind of like an engineering manual. You have the best big carp approach and the best match approach. The best big carp approach is to create sprayed pellets. So take your pellets of choice, spray them with 30 milliliters of liquid per kilogram, and then spot in about 7 spots. Not a massive amount, right? And then over the top of that, you fish those same dose pellets in a medium PVA bag, and that's really what got that 13-to-1 catch rate. So less is more sometimes, yes. You don't want to overdo the amount of impulse. You can do too much. So you can't throw in 20 spots. You can throw in 20 spots, just not dosed. A great thing to do is throw in your 20 spots and then 3 or 4 of dosed stuff over the top. You got to keep that impulse level perfect, even if you got more bait, if that makes sense. Yeah. Then we've got time for maybe 1 or 2 more. Here we go.
Can you use the bait soak? As in pop-ups, waffles, hot baits?
Yeah, it's not really recommended unless the baits are very porous. And there's a problem with pop-ups because by definition they're kind of watertight. Yeah, so what we'd like to do, we've done this with zigs as well. So we take the zig foam and we kind of basically absorb the water into the liquid impulse into the zig foam, and that works really well. What you probably want to do with a pop-up is either put some gel on it or fish it with a little bag of dosed pellets on the hook so it sits underneath. You want it doesn't have to be impulse coming from the bait, it's just the area effect. So get some pellets in the area and fish a pop-up over them. I often fish a plastic corn over dosed pellets, and there's no impulse in the corn, but I get a lot of fish. Yeah. 1 or 2 more? Anyone else?
Is it affected much by temperature?
Great question. So the answer is no. It's a very good question actually, because people often associate amino acids with proteins, of course. And when you cook an egg, proteins denature at cooking temperatures, don't they? So the egg yolk goes white. But that's the protein in its longer chain form. The bricks themselves, the amino acids, are super stable. You can go up past the boiling point of water before decomposition. So amino acids are susceptible to microbial action, but not heat. Yeah, it's a good one. Any more? All right. One more.
You said about spraying the pellets. How long after spraying would they be useful?
Great question. So think of food, right? So if the food is kind of warm or damp, it doesn't last as long, right? So what I like to do with my pellets is I actually spray them and 30 mils per kilogram, then dry them. And that's actually what a lot of bait manufacturers will do. They'll spray their pellets with flavors and colors and things, and they'll just dry them. And we've actually done that before, and they last a really long time. So at least 6 months dried. So yeah, so if they're dry, don't worry about it. Just use them through the whole season. But for me, I take a garden sprayer with me, and I spray my pellets on the bank, and they're wet, and and they're PVA friendly, so they go in the bag damp, right? And they just use that day. So I'm a bit lazy. I just use them on the day. Yeah. Yeah. Well, thank you for your attention, and come see me at the booth. Thank you.